My name is Michael McKernan. I'm the Director of Government and Community Relations at the Jackson Laboratory. I'm based in Bar Harbor, Maine. A brief word about the Jackson Laboratory, because as I travel around Maine and talk to people, neighbors, people in the state, organizations, we hear a lot of people who say, well, yeah, I've heard of the Jackson Laboratory, and I think what you all do up there in Bar Harbor is good, but I have no idea what you actually do. So one of the things that we'd like to do is tell you a little bit more about what JAX does as a research institution, and then really the focus of today's panel is a deep dive on the genetic basis of addiction. So the Jackson Laboratory is an independent, nonprofit biomedical research institution. Our mission is to discover precise genomic solutions for disease and empower the global biomedical community in its shared quest to improve human health. From its base in Maine, JAX is empowering the global biomedical community through education and, yes, through its mice. So a lot of people know JAX as a mouse house, and it's true. We have a lot of mice in our house, probably several 100,000 more than you do. But ours are more or less under control. Actually, I should say completely under control. I don't want to steal any thunder from my colleagues, so they're the mouse experts. I'll let them do the talking about that. But in addition to JAX, we're not the only member of Maine's really rich and strong research community, and this panel really couldn't have been teed up any better than was done by Ryan Hampton in talking about the importance of the science. We're joined in this biomedical effort by organizations like the University of New England, Maine Health Institute for Research, certainly the University of Maine, Wabanaki Public Health and Wellness, and many others. So several of these institutions have projects focused on the biological origins of substance use disorder. And while broad understanding of the biology of SUD is critical, research is increasingly focused on personal genetics and how my genetic background, your genetic background, all influences in subtle ways the way that we all experience different types of illness. That is also combined with environmental factors and provide an important context for how diseases manifest differently in every single person and how they must be treated differently as well. So we've assembled an expert panel here today to share some of the important work they've been doing in their own labs and centers. As a panel, we know this is a pretty heterogeneous audience, including some fellow researchers, maybe a physician or two who have deep knowledge of these topics and would very happily, have passed their high school biology class. Others, biology may be a far distant memory, and we're going to try to make sure that you're included in this discussion as well. And we have a microphone here, so please at any point, when the panel is over, we'll do Q&A. And the other thing is we have an opportunity for you to pose your question anonymously if you wish to, so you can log into Slido and post your question there, and then at the end we'll wrap those up and make sure that your question is addressed. Okay, enough from me. My pleasure to introduce our panel. I'll introduce them one at a time, and then they'll give their presentations. So our first speaker is Elissa Chesler, professor. Elissa is the Ann Watson Symington Chair in Addiction Research and is Senior Director of Integrative Data Science at JAX. Our second speaker is Jason Boubier, Senior Research Scientist in the Center for Addiction Biology at JAX. Our third speaker is Vivek Kumar. Vivek is an associate professor, also in the Center for Addiction Biology, and again, JAX. And our final speaker is Jake Byerly. He's a postdoctoral associate. Postdoctoral associate means that Jake is still in the process of his training and leading to a next step, which maybe he'll let us know what his goals are, so I don't invent them for you all. Anyway, Jake is a great example of that JAX is not only a research institution, but we're also an educational institution. So he is part of our community as well as many graduate students and even undergraduate students, graduate and undergraduate students too. So a lot to get through this morning, and we'll get it kicked off right away with Dr. Chesler.
Thank you, Mike, and good morning. How do I advance the slides?
Yeah, that's important.
Thank you.
Green arrow.
I was powerless for a minute. Okay, great. So I'm going to talk today about some of our findings in the genetics of addiction. We do these studies in mice, and then we try to understand what they tell us about people. We know that substance use disorders are among the most complex of disease. You think about the variety of substances that are out there that people are using, the trajectory, when they start using, how they start using, what their life experience is before using and throughout treatment and recovery. We know there's many, as we heard this morning, predisposing environmental, developmental, and genetic effects that influences a person's trajectory. And mouse genetics give us tools to understand the complex relationships among genetics and behaviors of many different kinds and different psychiatric disorders, et cetera. Mice share over 90% of our genes, and genetic analysis that we do in mice can actually help us understand what is happening in people. We can look at how the genetic findings that we have directly compare to the genomes of individual humans and try to understand from existing data, cellular assays and other things, how those results in mice relate to what we know from humans. People, we call them people. Human people, all of us. So one thing we know is only some people who use drugs become addicted to them. It's very important not to conflate the use of drugs with substance use disorders. Almost 70% of people who use nicotine will develop an addiction to nicotine. For alcohol, it's somewhat less. And for other drugs, even cannabis, which has been perceived as largely harmless, people become addicted to. And we know that the interplay of genes, environment, and development interact to shape individual outcomes. So we heard about adverse childhood experiences. We've heard many different things coming together. So if you will, you're like a marble rolling down a hill. You can be pushed in either direction to end up somewhere on the spectrum from completely, or I should never say completely, resilient to substance use disorders to highly susceptible to the substance use disorder. Those of us of a certain age might remember better and relate to Plinko on The Price is Right, so you're like a Plinko chip. But I want to correct something I just said very clearly. Anybody with enough exposure can become addicted to a drug. So biologically not very susceptible, but still vulnerable to repeated use through environmental experience or just a consistent pattern of use. We know that young people who start using early, and we heard that the life experience can play a role in when people start, young people who start using early are most likely to develop a substance use disorder. So this is a chart of the age at first use. So the gray bar, the height of the gray bar is the age at which someone first uses. And the black bar, the black shading over that bar, tells you what proportion of adults who started using at that age develop a substance use disorder. So you can see it's for nicotine, alcohol, other drugs. It's very likely if someone starts using before the age of 14 that they're going to develop substance use disorder after the age 18. We know some things from mice too. We know that the complexity of the environment influences how likely a mouse is to engage in substance use disorder. Mice that are reared in complex environments are, well, we don't call them substance use disorders in mice, but mice that are reared in complex environments are less likely to intake drugs. We don't know if they're using them. Mice that are exposed to nicotine in adolescence are more likely to self-administer cocaine in adulthood. And this is an experiment that we did in our lab recently, where we asked the question, I was trying to find out what happens, like how do mice spend their day, and how does their day change if they become frequent, self-administer drugs frequently? Does that alter their normal behavior patterns? Do they stop making a nest? Do they stop doing other things? And if I take the drug away, are they going to go back to doing all the things that they did or develop a different compulsive behavior or basically hang around looking for the drug? Anyway, I couldn't do the experiment. We found that when we put mice in a complex environment, like just with other things to do, they simply didn't acquire drug self-administration. And so we said, okay, let's put them in the box where we normally do these experiments and say, oh, see if the mouse can be trained to self-administer drug. They do. And that's what's shown in the leftmost panel. And if we then move them to their home environment where they have things to do, they completely stop self-administering the drug. And if I then put them back, I say, well, maybe they just extinguish that behavior and it's all good. Let me put them back in that kind of impoverished environment again, nothing to do but drugs, and they go right back to the level of drug intake that they were at in the more complicated environment. Now, we don't want to anthropomorphize too much. People aren't doing all the same things that mice do, but we can see here that given enough other stimulation, given enough other options, the mice are less likely to self-administer the drug. So, as I said, it's not the whole story, but genetics can explain a large amount of addiction vulnerability. So if we look at cocaine here, 70% of twins exposed to cocaine are likely to share a cocaine use disorder. Similarly for opiates and other drugs and even gambling, there's some fraction of heritability to substance use disorders. And when we look at mice, these are 8 different strains of mice, and every color represents another strain. These are perfectly inbred, so every mouse, it's not like twins, it's panels of clones. Every purple mouse is the same as every other purple mouse. There's 1 dot here for males, there's 1 dot here for females. But what we can see is that all of these other behaviors, their novelty response, risk-taking, impulsivity, self-administration, the sensitization of cocaine, how much they're acutely influenced by individual doses of cocaine, and even the circadian rhythms, which we know are a risk factor for substance use disorders, all of them vary in genetically different mice. So the mice that are genetically the same are more similar to each other than they are to other mice. And you can see that there are some strains of mice. If we look at the lower right, there's a lot of jargony abbreviations there. But in the middle of the bottom row, we see how likely mice are to infuse cocaine, how likely they are to take a dose of cocaine. And we can see the red strain, very high self-administration of cocaine. Other strains like the purple one and the pink one, not so much. And sessions to acquisition, that's how rapidly they develop that repeated behavior. And we could see again that red strain of mice, PWK we call them. PWK very rapidly acquires self-administration, whereas most of the other strains not so much. We also see that gene expression in the brain differs across these mouse strains when we administer cocaine, but also just how they are, how they come into the lab before they start using the cocaine. So we looked at, you could see that the patterns of gene expression, I'm not going to go through all the math here, but on the upper left we can see that gene expression patterns in the brains of these mice is enough to classify them into these different groups. We could actually tell the mice apart by what's happening in their brains. And we can see that in terms of the response to cocaine, the number of genes that are influenced by different factors, sex, differences, male or female, the strain of the mouse, and that exposure to cocaine. The number of genes that are influenced by cocaine exposure most of the genes that are influenced by cocaine exposure interact with sex differences in genetics. So the biology of the individual plays a major role in how the brain responds to cocaine. And this is just on the bottom, an example of one single gene and how the response to cocaine differs across different strains of mice. Here's another interesting thing. Exposure to drug, learning that self-administration of that drug, so repeatedly self-administering cocaine, creates lasting changes in brain gene expression, and that effect is different in different strains of mice. So the strain that we'll call NZO, New Zealand obese, I think, NZO stops drug seeking. So I'll go back here. On the left, this is the baseline. They come in, they don't have drugs yet. In the middle, they've self-administered the drug. And then on the right, we've taken the drug away. And if we look at NZO, stop seeking the drug when we take the drug away. They walk away, they leave it alone. And the expression of a group of genes goes down in that strain of mice. But if we look at CC19, we take away the drug, and CC19 mice are still looking for it. They're pressing a lever even though nothing happens. And in this strain of mice, the set of genes that got turned on while they learned to self-administer the drug doesn't turn off. They have this lasting expression change in their brain. And so what is that? We know that there are what we call epigenetic changes, switches in the genome that can be turned on and off to turn on and off in a lasting way in response to experiences, what genes are expressed, how often, where, and under what circumstances. And using genetics, which is the left is genetics, and the right, analyzing all that genetics, we can pinpoint where in the genome are these switches that influence gene expression and behavior. And so there's an example of one that's on mouse chromosome 7 we found here. And using other methods, genomic experiments, we were able to see that the variant that we found on chromosome 7, the difference between different strains, one or a few, we don't know yet, base pairs in the mouse, is at the location of what's shown in green at the bottom, one of these genetic switches that regulates 3 other genes, 2 of them highlighted in yellow, but connected to that green switch. You could see the loops. Those are the other genes that are turned on and off by that switch. And I told you, one strain of mouse has a good on-off switch. We think the other strain of mouse has a broken switch. We don't know if it's locked on or locked off. It's still called research. But when we then look at that switch and we say, is there another switch in people that turns on and off the same genes? And yes, there is. And so then we took that switch and we said, are there any differences in people at the location of that switch, where one set of people might have their switch stuck on too? If that switch is broken, the individual response to the environment might be locked on, it might be locked off, and that might lead to a predisposition to the same drug-seeking and drug-taking behaviors, and even the baseline risk-taking, novelty-seeking, and other behaviors that we see in mice. And so we looked at a lot of other genetic mapping studies that were done in people, and whether that location influences these traits in people. And you can see on the bottom row of this is quite a number of them, both predisposing conditions that are frequently co-occurring with substance use disorders, but also a number of different characteristics. Some substance use disorder, computer game use. You know, we talk about kids in social media and their addiction to the cell phone, but there's actually genetic predisposition to get hooked even on electronics use. And so we can find these places in the genome and then try to understand what to do with this. So in summary, susceptibility to substance use disorders varies among individuals. They emerge from gene environment and development interactions. And using mouse genetics, we can find some of the mechanisms by which this is occurring and examine how they inform what we do and how human genetic variation influences the trajectory of people. Lots of people work with me, and they do great stuff.
Thank you, Elissa, for that introduction. Now I'm going to tell you a little bit more about the actual cause of death that we see with substance use disorder, at least with opioids, is the opioid-induced respiratory depression. So Elissa showed you about the heritability of your addiction to different compounds, how likely or how much your genetics contributes to your susceptibility. It turns out genetics also contributes to your susceptibility to overdose. So that's why there can be families where all three children in the family all overdosed because they were all an equally sensitive background. So the genetics can explain part of the sensitivity to overdose. And so I'm going to talk about how we're going to be able to use mice to understand this. So as you know, there are multiple functions to opioids. They work for the reasons that we often want to use them is for the analgesia, but the side effect that we see is the respiratory depression, the cardiovascular depression. A lot of people who've used them know that they result in decreased gastrointestinal motility or constipation. Sedation occurs, the euphoria, why people may abuse or develop their substance use disorder, as well as urinary retention. So the opioids can do many things. I'm going to be talking about that aspect of which the opioids are working on respiratory depression. A quick little basis of how opioids work in the brain. So as I mentioned, opioid overdose, which you reverse, hopefully, is through the opioid-induced respiratory depression. There's these receptors called the opioid receptors in the brainstem, specifically in a region called the medulla, that contain cells that control and respond to concentrations of like oxygen and carbon dioxide and determine how fast and how much you should be breathing. And it's opioids binding to those receptors that can cause the depression in your rate and result in death. Now we have this great miraculous cure that's been developed, naloxone, which is developed as kind of is the antidote or the overdose reversal agent, and it's going to work in a replacement mechanism. So now in the little picture here, the opioid is the little red circle, and then the naloxone coming in is the group of green circles. And what happens is that it's going to develop affinity or bind strongly to the receptor, pop out the opioid, and instead it's going to go through there and stop sending the signal. So there's a signal coming through the receptor when the pink thing is on there, but when the pink thing is off and blocked by the naloxone, there is no more signal and your respirations can return to normal. So this is how naloxone works. And as it was mentioned earlier by Ryan, one of our concerns right now is the addition of other drugs such as xylazine and tranq is resulting in the ability for naloxone not to work because the tranq involves xylazine, which works through a different pathway than the opioid receptors. So something else is going to be needed, and maybe it's a supplement to naloxone to help in those situations. All right, so Elissa mentioned that mice are similar to us. About 92% of our genome is the same. Within people, our similarity to each other is 99.9% similar, but as you can see from these minifigs, we all look very different. And within that differences we see physically, there's a lot going on genetically and in our DNA that manifests in this way, and that's how we show this difference. And a key thing that I want to point out, or my work is showing us, is that we can use the genetic diversity of mice, so shown over here with different colored mice on the left, and the genetic diversity of a human population shown by different groups of minifigs on the right, to help us find regions of the genome that control susceptibility. So those top group of minifigs, those people need high doses of an opioid in order to get the alleviation of the pain, as well as in order to, they can resist a dose and they will not die from overdoses easily. The ones in the middle are middling, they have a middle dose, and then the ones on the bottom are very, very sensitive. So a little dose in one on the bottom, it would, 50 times the dose at the top to overdose on those people. So we don't know, looking at them, that it's, because you have the blue face or the gray hair, that you're going to have which sensitivity. But we can, by looking at our DNA, and that's what the arrow is pointing to, is that region in the DNA helix, that might correspond to that region. Explaining that, controlling that sensitivity. So why are some more sensitive? So here I'm going to talk about two strains of mice. We're going to call one a sensitive strain at the top, and this actually happens to be the strain that Elissa told you about was very happy to take and use cocaine. So that's an interesting polydrug phenomenon. It's also very sensitive to fentanyl. So this is saying that its LD50 is about 2 milligrams, whereas another more resilient or resistant mouse takes about 50 times that. 100 milligrams. So it can use a big dose, whereas the top mouse, it can only take a little dose. So then I have 3 plots here where I look at a couple of the characteristics of a response to the opioid. So I give the mice fentanyl, but first I measure their number of breaths in the top left. And so what you can see is that the resistant strain is breathing a lot, high frequency, and the sensitive strain has a less frequency, and this is without any opioids. This is their natural baseline respiratory rate. And then if you look at the second plot, it's their depth of their respiratory breaths. So are they big breaths? Are they breathing in big, or are they shallow? And you can see that the resistant or resilient mouse has more shallow breaths than the sensitive mouse. And then if you go over to the final figure on the far right, on the bottom, this is looking at the arterial blood gas. So this is the gas coming out of the lungs after it's been given its fresh dose of oxygen, and how much oxygen is dissolved in the blood. And the resistant mice or resilient already starting this game with a more oxygenated blood than the sensitive strain. The sensitive strain is starting the game with a less oxygen capacity in its blood. And now there's going to be 3 time points, 15 minutes, 30 minutes, and 45 minutes after giving them fentanyl. And it's the same dose of fentanyl, but we know there are different sensitivities, but the same dose given to all the mice, and it's based on weight. And what you see is that in the top figure, the depth of the breath, all the mice have respiratory depression, so it really goes down. And this is what you're trying to reverse when you use naloxone, is the depression of the breaths. And you can see that the sensitive goes down a little bit more than the resistant, but it was already down a little bit more to start with. But the interesting thing about this sensitive strain is it's trying to compensate in the bottom by increasing the depth of its breaths. So those mice, if you can see in 30 and 45 seconds, their breaths are much deeper in that sensitive mouse. And even though their breaths are much deeper, but fewer, if you look at their blood gas, they've really plummeted. Their blood gases have gone down. Their oxygen levels are very low. And so when their oxygen levels are low, then their carbon dioxide levels are going to be high. And this is what's going to result in them being unconscious and then dying. So what we're able to do is use the mechanisms of variation in a population, here variation in a mouse population, to understand physiological basis for sensitivity to opioids, and with additional physiological risk or their initial assessment of how they respire to begin with, to predict how they're going to possibly respond to drugs. So one of the things that this results in is, in the figure, I talked about how respiratory depression and naloxone generally works for reversing the frequency of the breaths per minute. But over on the right, you can see that, and if you remember back into your high school biology, that there's a level of gas exchange happening at the lung, where the oxygen and the carbon dioxide are exchanging with the blood cells. That's a level that we don't target with any of our respiratory depression agents, but could be another place that we could target. So if we wanted to look for a reversal agent that increased the oxygenation of the blood, that increased the permeability at that barrier, and we could maybe look at other compounds that are being used for other diseases to see if they could be added in combination with naloxone to increase the response. And this is especially important when these other second and third generations of drugs like carfentanil, the elephant tranquilizer, that one is synthetic, designed in a lab to bind so tightly to that opioid receptor that it never really wants to leave. So it's very difficult to displace it, and it requires sometimes these multiple 3, 4 doses of naloxone to reverse it. So what I'm trying to illustrate here is that using the basic biology of the mouse and the diverse population, I'm able to look at different mechanisms of action of these drugs and possibly point us to new areas to look for targets. Instead of targets involving the opioid receptor, maybe we need to be looking for targets involving gas exchange. Finally, if we do find that region of the DNA that's different between the different strains, and we can find that same region in humans, as Elissa said, if we're finding that homologous, orthologous region in humans, we can use it as either for a variety of different reasons. We can use it as a biomarker. Before we prescribe anyone any opioids, we can determine, are they going to be needing a high dose or a low dose of opioids? It's called pharmacogenetics, where your prescribing is based on your genome or your personalized medicine, where you decide, okay, this person needs a low dose, this person needs a minimum dose. So that's kind of the predictive aspect as well. Safety, again, and risk. All these different things can be used in arrays that are done for genotyping. You might be familiar with like 23andme.com or ancestry.com. They do broad genotyping. If these alleles are identified, they can be included in such arrays, and then everybody for $100 can figure out what their own sensitivities are, as well as through the medical field. So my takeaways from this, and I didn't really go off on this at the beginning, but Ryan mentioned this as well, is that addiction is a disease, and it has a biological basis, and it is not a moral failing. And that's like the number one thing that I try to tell my 84-year-old dad every time I go home, and he says, "What do you do again?" And he says, "Oh, they're just not strong enough." And I'm like, "Oh, no. They're given a different deck than you are, and in that deck can include their life experience as well as their genome. And so it's not a moral failing. Like addiction, opioid overdose has a biological mechanism, and each person has their own potential or risk sensitivity. You can use the genetic variation in a population to identify new targets, to develop new reversal agents, to combat the current and emerging threats. As I mentioned, the different combination of drugs and the newer, stronger synthetic opioids. And that model organisms give us the ability to try to separate the different functions of the opioids, separate the pain relief from the respiratory depression, from the gas exchange. And if we're able to understand these biological pathways, we can target them differently. And that's it. I'll cue it up for Vivek.
Thanks, Jason. Thanks, Elissa. Thanks, Mike. I want to thank Governor Mills. This is, I think, the third or the fourth summit that we've presented at, and it's a really special event, and I think people of Maine are really fortunate to have this advocacy at the top levels. So I'm going to tell you about something related to addiction and substance disorder, but still different. So we know that substance use disorder is not an event that happens to individuals by itself. It's often comorbid with a lot of many other sort of neuropsychiatric conditions, even neurodevelopmental conditions. So things like schizophrenia, bipolar disorder, anxiety, even autism and other sort of neurodevelopmental disorders can be predictors of addiction. And in fact, substance use disorder can be a form of sort of a symptom rather than the cause of some of these events. So the main question that we're interested in answering is using the mouse to model these complex neuropsychiatric traits. And my lab is focused on not just addiction, but really understanding how do you use the mouse in a really better way to understand mechanism and to get at treatment for human conditions, especially these sort of abstract things such as neuropsychiatric conditions. So it begs the question, right? How do you study like the emotional state of a mouse? What is a stressed mouse? What is an anxious mouse? What does a mouse model of autism look like? What does a mouse model of bipolar disorder look like? What are the behaviors that change? We unfortunately can't ask them how they feel, so we have to sort of derive the internal sort of emotional state of these animals. And this is actually a tough task, and in fact, I would argue that scientists are not great at understanding the emotional state of at least mice. I mean, when you look at your pet, like your dog, like you could probably tell how it's feeling, like you have a connection to it. But in a laboratory setting, it's really hard to determine the internal state, the emotional state of an animal. And sometimes, unfortunately, we blame the animal for a lot of the problems of our sort of studies. So we take an approach that's different, but it's an older approach of behaviorism, which is called ethology. The idea here is to study just naturalistic behavior of mice or animals. This is Jane Goodall. She didn't study chimpanzees in a zoo. She went to the jungle, to the wild where they lived, to study chimpanzees. So she went and studied animals in sort of their more native habitat. So we try and do something similar with mice. Obviously, we can't go to the field and study mice, but we just look at mice in their sort of natural environment, which is in our lab is a cage or a larger sort of arena, and we try and understand from how much it's interacting with animals, how much it's eating, drinking, feeding, all these sort of events that the mouse carries out, what is its sort of internal state, like its emotional state, and link it to diseases. So how do we do this? So the way we do this is obviously humans are the best in terms of observing animals, but we can't have a trainee or a technician look at animals all day. So we actually train artificial intelligence and machine learning methods to do these. So we have these cameras that we watch the animals with, and then we have really fancy computer algorithms, machines that are making predictions on what the animals are doing. So here we have a single animal on the left or multiple animals on the right that are being tracked in this open field sort of large arena. Then we can watch how much it interacts with its friends, how much it fights, how much it mates, how much it eats, drinks, all these different things. This technology that we've been working on for almost 10 years now has really sort of taken off in the field, and the Jackson Laboratory has turned it into like scalable platform. So now people all over the world can use these technologies. This is just kind of one of the racks that we have, where we have 48 of these cages with cameras attached to it, and it collects really high quality data like the one you see on the left and the right in the daytime. The one on the right is actually filmed at night. Mice are nocturnal, so you really need to observe them in the dark for interesting behaviors. And we operate on data like this. Okay, so I want to show you just one example of how we use these methods to understand human conditions. So Down syndrome happens in about 1 in 700 births. It's a neurodevelopmental disorder. The main thing in the clinic with Down patients that we want to work on are things like intellectual disability, kind of cognitive traits. However, if you're a parent of a Down child, you know that that child hurts themselves. They'll fall. They're sort of clumsy children. And we know like motor coordination, like changes in movement, is sort of a hallmark of changes in the nervous system and neurodevelopmental disorders. So the question here is, could we model movement, like could we use movement as a surrogate for higher order sort of cognitive function? And to do this, we're going to look at a mouse model for the study of autism that was also developed at the Jackson Laboratory. And the original work in this mouse model was done to look at motor coordination deficit was done by a previous faculty at JAX, Muriel Davison, and she looked at sort of these motor coordination deficits by putting ink on the back feet of mice and having them walk on paper. And if you look on the left, the control mouse sort of has a straight pattern, and the mouse on the right has a slightly sort of crooked pattern. And that's evidence that the mouse has motor coordination deficits. So what we did was we took this same mouse model that she designed, and we put it into our open field, and we tracked different body parts of these mice using our AI ML algorithms. And now we can really collect lots of data, and we're not forcing the mouse to walk, it's just doing what it likes to do. We're just putting a camera on top of the arena. And from this, we derive something like this. So this is that same image of the mouse with key points on it, but we're just kind of overlaying thousands of them. And the point on the very right is the nose of the mouse, and the point on the very left is the tail tip of the mouse. And if you watch sort of that third point from the left, which is the base of the tail, so like right where the tail touches the butt, it kind of moves when the mouse is walking, it moves up, it'll come back down, and it'll sort of end up in the middle, right? So mice are not walking in like a straight line, they actually sort of have a swagger, right? Like they've got rhythm. And so we looked at that, and you can actually quantify that in using sort of mathematical models or fitting of data. And we can look at sort of the phase and the amplitude of that movement of that base of the tail, or we can call this like the posture of the mouse. And if you look in the down mice, which are in the dashed here, they're completely different than the controls, right? So here it is, this is a mouse model that's been sitting on the shelf for 20 years. We're interested in fixing or using it to understand mechanism and develop therapeutics for a cognitive trait. And by studying, but the cognitive traits are really hard to study. You have to train mice for months, weeks, months to really perform a cognitive task. But here we could use a motor task as a surrogate for that cognitive task and screen hundreds and thousands of therapeutic compounds using this approach, at least theoretically. So that's the idea. I'll also just kind of mention that to do this work, we are really a multidisciplinary team. Like at the Jackson Laboratory, we have neuroscientists, we have geneticists, we also have AI/ML experts, we have computer scientists, we have statisticians working together, and all of our work is funded by NIH. I think the keynote speaker mentioned the need for more kind of continued research funding, and I think the funding from NIH has enabled a group of us, really good, important people, but still on an island to do just really meaningful work in Maine. So I'm always appreciative of that. All right, thank you.
All right. Perfect. So I'm Jake Byerly. I'm a postdoctoral fellow in Vivek's lab, and I'm going to continue on the same sort of theme, but give you two discrete examples that we're using in the lab, or that we're applying this technology to in the lab that are related to the opioid crisis, and that's chronic pain and opioid withdrawal. I'm really grateful that we had such a good introduction and a good speaker, right? Because I don't feel like I need to justify why chronic pain is related to the opioid crisis. But just to summarize, oh yeah, sorry, they asked us to put a disclosure statement in here. No financial disclosures. Opioids, chronic opioid pain management comes with the risk of opioid addiction, right? Which raises the question, why are we still using opioids to manage chronic pain? Right? And beyond the fact that opioids are actually very useful analgesic drugs, the other answer is the drugs that we have that are alternatives to opioids come with serious compromising complications, right? Several of these drugs, so these are 5 very common classes of drugs that are used to treat pain. Some of these drugs have limited maximum effects, so they don't provide enough analgesia. If you have a kidney stone and I give you aspirin, you're probably going to be frustrated with me, right? They either come, or they can come with really troublesome complications with chronic use, right? So corticosteroids compromise your immune system. They decrease your bone density, which is a serious problem, particularly for older women. And these local anesthetics are really short-lasting pain relief that aren't well equipped to treat chronic pain in patients. And so the consequence is a lot of physicians have turned to opioids to manage patients' chronic pain, right? And while opioids don't come with these really troublesome, immunosuppressant problems like corticosteroids, they do come with other serious complications, right? Primarily, the longer you are on an opioid, the more opioids it takes for you to get that pain relief that you're seeking, right? The therapeutic effect of the drug. And that's kind of illustrated in the blue curve on the left and the red curve on the right, where to get the same amount of pain relief, after chronic use, you're using much more opioids. And escalating your opioid dose puts you at increased risk, not only of overdose, like Jason discussed, but substance use disorder. So why then are we having such trouble developing chronic pain medications? This is a schematic that's showing you sort of the drug approval process from the most basic steps on the left to a single approved drug on the right. And what I wanted you to focus on is that third preclinical test phase, where it takes about 10 to 20 compounds to be tested in animal models to yield one usable drug in humans. And that rate is actually, there's a 5 times increased failure rate for analgesics than the average drug, right? So it's not actually 10 to 20 analgesic drugs that have to get screened to yield one usable analgesic drug. It's about 50 to 100, right? And so the question that I'm asking and that Vivek is also asking in the lab is, are we identifying the right candidates in our mouse models, and how can we improve the way that we screen these drugs so that we can really select the appropriate compounds to move forward with in patient populations? And one of the reasons that we think we're having trouble moving from mice to humans is because we use fundamentally different tests in mice than we do in humans to assess pain, right? So this is a sensory test that we use in mice. It's a very thin filament, and we use it to apply a very light mechanical pressure to the bottom of the mouse's foot. And depending on how much pressure it takes for that mouse to withdraw its foot, we sort of infer its relative pain state. But as anybody who's been to the doctor for pain knows, when you go to your clinician and you say, I'm in pain, they do not pull out a Von Frey filament and poke the bottom of your foot. They simply ask you, on a scale of 1 to 10, how much discomfort are you in? And so the goal of the work that we're doing in lab is to move away from those artificial mechanical stimulations and move closer towards this sort of scale where we can infer the psychological state of the animal instead of relying on a sensory test. And so the underlying hypothesis that we have is that by using these voluntary behaviors, ethology like Vivek just said, we can better model patient-relevant outcomes, and we can have higher success rates at lower cost when we develop analgesics for patients, and hopefully divert patients away from opioids and remove the risk that they transition into a substance use disorder. So the way that we do that, like Vivek sort of discussed earlier, is we let a mouse voluntarily behave in what we call an open field arena, which is essentially a box, right? And as the mouse behaves, we use a machine learning algorithm to tell us where its head is, where its tail is, where its paws are, and where its spine are. And the way that these points change over time and in relationship to each other and in relationship to the arena, we can get a really rich index of behaviors that we can then use to try to understand the way that the mouse is feeling, right? And these include some of the traditional measures that have been used in the field for a very long time. We can classify behaviors that we're interested in, like grooming and exploring on its hind paws as it stands up, and freezing. And we can also extract those gait and posture measures that Vivek was discussing in the Down syndrome model. So I've applied this principle to a modality of pain called neuropathic pain. It originates when you injure peripheral nerves that encode sensory stimulation, so mechanical, thermal, proprioceptive stimulation. And the reason that this is a very interesting pain condition, at least in the terms of opioids, is it is very poorly managed by opioids, but does not have a lot of other particularly efficacious drugs. There are first-line treatments that are not opioids, but a lot of patients get diverted to opioids and have that really severe dose escalation because of poor pain management. So we want to develop better measures in this model so that we can screen compounds that have potential therapeutic efficacy that allow us to move away from opioids. So what I'm showing you is kind of a scary figure, but essentially what each column is a time point that I'm observing the mouse's behavior, and each row is a behavior that we can extract using our sophisticated machine learning tools. And the darker color, either red or blue, of those tiles indicates a more significant disruption in that behavior following neuropathic injury. And as you can see, we've got groups of behaviors on the top and the bottom that represent very stable disruptions in the mouse's behavior after injury and during neuropathic pain. And then in the middle, we have these dynamic changing behaviors that probably represent some sort of functional recovery or a dynamic change in the mouse's psychological condition. And so we can now use this as a baseline to understand how drugs restore mice back to their normal behavior and use that to infer pain relief. The other thing that I want to really briefly talk about, a more nascent project, is using these same theories or these same approaches to quantify opioid withdrawal. Right? Because opioid withdrawal, as we kind of heard earlier, is one of the primary maintainers of opioid use, at least while people are initially trying to quit, right? It's very unpleasant. And within mice, much like cocaine acquisition and respiratory depression, different strains of mice have different withdrawal severity. So what you're looking at right here on the left is a mouse with no withdrawal. It's ambulating, it's moving around, it's exploring the arena. In the middle, we have a mouse with low withdrawal phenotype where the mouse is going to freeze. It's a prey response. It's trying not to be seen by an aerial predator. Right? And on the right, we have the same mouse strain on the left with a high withdrawal response. And this mouse is trying to escape the arena by jumping. And then within those bouts of jumping, the mouse is freezing as well. So what we propose to do in this particular project is take those videos where we use AI to extract behavioral features. This is a list of punitive features that are very well described already within the literature that we can automate and extract, and then combine these features to make a more representative withdrawal score that we can use across genetic diversity to try to understand how genetics interacts with withdrawal severity and identify biology that we might be able to target to relieve withdrawal as patients try to cease opioid use. And with that, just to kind of summarize, we really think that these techniques allow us to access more of the psychological state of the mouse and move away from reductionist assays that by necessity have been adopted before. We're moving towards a more enriched context, as Elissa alluded to, that allows us to look at behavior in a social context, because humans tend to use drugs in a social context. And we really do think that these approaches are really ideally suited to interrogate opioid use disorder behaviors and their related comorbidities and public health crises. So thank you, everybody, for your attention.
All right, thank you very much. Excellent panel, lots to think about. We have a couple questions that have already come in via Slido, and I'd invite others who are considering asking a question to go ahead and use that, or again, to come up to the microphone in the center of the room. So the first question is related, I think Vivek will start with you on this one. So the question is, how do we know all identified, in quotes, Down syndrome mice are actually arthritic mice, for instance, and thus all the results discovered regarding Down's mice are actually results for arthritic mice? So how do you sort of exclude these other potentially confounding factors?
Yeah, that's a great point. So this is where we rely on that precise sort of mouse model that's been created. So that mouse model that Muriel Dobson created, she's taken, so Down syndrome is trisomy 21, so it's the 21th chromosome of the human genome that's duplicated essentially, so you end up with 21 chromosomes. So she took the same region of chromosome 21 in mice, that's called a synthetic region, and she essentially created a duplicated model. So she, in the mouse, created a duplication of the region, chromosome 21, synthetic region of chromosome 21. So we're relying on that. So when we study, for instance, mouse model for the study of autism, we take mutations that are known to cause autism in human cases, and recreate them in the animal. So that becomes sort of our, our sort of avatar type mouse, or like a mouse model for studying these things. Yeah, but we can also look, if you're interested, like a lot of neurodevelopmental disorders do have comorbid conditions. So we could easily look, for instance, at joints and functioning of muscles and bones also.
Got it. Thank you. One of the things that's amazing at JAX is our ability to measure almost anything that could potentially be designed and measured in a mouse. It's rather remarkable. So the next question is, as researchers and practitioners, what is your opinion on the fairly new non-opioid pain relievers coming out in the market? Also, any thoughts on or opinions on non-drug procedural pain management, i.e. epidural steroid injections, et cetera? So I don't know who wants to take that on.
Yeah, so I think that there is a valuable move towards non-opioid therapies like physical therapies, right? Combinatorial therapies. And there have been newly approved sodium channel blockers that are used for pain management right now. It's a really interesting philosophical topic when you think about what makes pain and a good pain management drug. Anything that fundamentally blocks incoming signals is not necessarily what you want, because pain is very informative and helps you preserve your own health, right? And what you really want to do is probably alleviate the associated discomfort and negative emotion that comes with pain. So it'll be really interesting to see how these sodium channel blockers evolve as they get prescribed and how efficacious people find them to be. I think there's still, room and demand for new drugs beyond that don't just block the encoding of noxious stimuli, because that is really important information for an organism and, humans.
Thank you, Jake. So I'm going to address the next question to either Elissa or Jason, and you can duke it out. Earlier, we heard about correlation between adverse childhood events, ACEs, and addiction. Does research show that ACEs are able to induce epigenetic changes in specific genes that affect addiction susceptibility or behavior regulation? And you might need to define epigenetic.
So the answer is yes, that indeed life experiences can influence later vulnerability to addiction. Some specific epigenetic mechanisms have been identified. Transgenerational inheritance has been seen. Some of the populations people have looked at are people that survived the Dutch hunger winter, classic studies on that, those individuals that had starvation during that period. There's another in China where there was a large famine, where those populations had increased rates of substance use disorder, vulnerability to schizophrenia, and a number of other conditions. People have looked in epigenetics from a number of populations that have experienced trauma, and there are indeed increased epigenetic effects of those life experiences. What we haven't done in detail is connect those specific events to specific vulnerability to aspects of substance use disorder. So it's naming them, identifying, understanding what the genetics and gene expression consequences are, and then further understanding which of those might be harnessed to identify approaches that can be used to facilitate recovery in people who have that biological difference that's been shaped by the environment. So we can ask the question, for example, by studying mice, what's the biology of the mice that are able to stop seeking the drug when we take the drug away? How does that differ from mice that continue drug seeking? And can we induce the state that we see in mice that are able to stop using the drug? Can we use drugs, behaviors, other types of interventions to get those mice into a condition where they're more likely to stop that drug-seeking behavior? Good question.
All right. Yeah, these are all great, and the questions are coming in, so it's exciting. So, Jason, you mentioned xylazine tranq. The question is JAX currently working in collaboration with or in a joint venture project to create an agent to combat xylazine or tranq, is the question.
Currently, we aren't funded to do that. I have written proposals that haven't been funded yet to investigate this, but I am working with other collaborators, Elliot Nelson and Arpana Argawal at University of St. Louis, Washington, WashU in St. Louis, and they're doing human studies, and they have a grant under review right now where they're looking at single cell transcriptomes of xylazine exposed and opioid exposed compared to just opioid, compared to just controls. So kind of a three-way comparison to look at the changes in the genome that they're, and I'm looking at it from the mouse angle, and they'll find things in humans, and then I'll look at them in mice, and we'll go back and forth together. And so hopefully that one is under review and will be funded.
And if that's funded, is that a multi-year study that's been proposed?
Oh yeah, it's pretty expensive because it's a human study. So getting these samples, people have to generously donate them post-mortem, and then it's a smaller sample size. But a lot of these genetic technologies are very expensive. So trying to do single cell on large populations, it's 5 years.
Right. Yeah. Just really underlines the importance of that continued support. An interesting question has been asked here. Has anybody studied alternative medicine options? And acupuncture has specifically been mentioned. But are there thoughts or studies in any alternative medicine avenues?
I am not aware of something like acupuncture or other alternative medicines. I mean, there are a lot of natural compounds that are studied in mice. But yeah, that's a tough question. Yeah.
So Jeff Mogul, who's I guess pioneered the genetics of pain in mice. I know there were some earlier studies that predate him, but he did most of the foundational work in the field. Did have a collaborator, Wan Yu from China, who was studying acupuncture, and I don't remember the results of those studies, but using this notion of looking for strain differences, how different mice respond to the intervention, he did look at acupuncture effects in pain management.
So a couple more questions here. What are your thoughts on the hyperalgesia in chronic opioid use? And I don't know what hyperalgesia is, so then more pain. Okay.
Yeah, so for those who don't know, hyperalgesia is this counterintuitive sort of paradoxical effect where as you continue to have prolonged opioid exposure, opioid goes from managing your pain well in the short term to actually eliciting more pain in the long term. This is just another reason to move away from opioids for pain management. I didn't really get into it because we're kind of focused on the substance use side of things, but it is another counterindication and danger of using opioids, that not only do you put patients at risk for developing opioid use disorder, but you no longer manage their pain efficiently on top of that.
Great, thank you, Jake. Okay, so final question here. I saw that mice share 90% of our genetics. I think it might be a little higher than that. But have you found any trends in mice that are not applicable in humans in terms of drug abuse patterns?
It's a little bit of a stumper. I would say I don't know that we've found major differences. I think what we've encountered are challenges that Vivek and others have been working to address, which is a lot of what we know about addiction comes from this paradigm of putting mice in a box, operant behavior. They press a lever, they get a dose of the drug. And so we're looking at these very early responses, and we actually, receiving that drug was termed reward. And we named the brain circuitry the reward circuit. And so we've reduced addiction to people trying to collect too many prizes. And I think a challenge is, we know there's very little rewarding about getting a drug when you're fully addicted to that drug. It relieves your withdrawal. It may, it's more of a compulsive behavioral drive. And so we have to be very cautious about how we interpret mouse models, what we say about the behaviors that we think we're studying, and how we label what we're doing. I'm looking at mice probably in the first 60 days of them pressing a lever for drugs. And we know that, and that's a longer part of their lifespan than it is of ours, but it's still not that long-term chronic pattern of repeated withdrawal and repeated attempts to quit using drug and the increasing compounding challenges of having a substance use disorder. And so it's very difficult to model those things. And we think some of the technologies like Vivek is using, some of the approaches that we have to directly, computationally, integrate data from mouse and human genetic studies are ways that we can create better interpretability and understanding about what the results of our experiments mean. And again, really be cautious about how we're interpreting what it is that we're studying. How do we study that locked in behavioral drive, the behavior of getting stuck in repeatedly using a drug, spending most of your mental energy trying to find that drug, obtain that drug? That's not something that we can easily study in mice, but we can understand what it is that we do in mice that relates to what it is that's happening in people by evaluating the similarities of these genetic findings that we have.
Well, the search continues. Always more questions to be asked and answered. The need is tremendous. And Maine, and I think, just speaking as your colleague, I'm personally, express a feeling of great fortune that we have the minds at work that we do in the state on this tremendous challenge. So thank you all. Thank you all.